Welcome back. Last time we talked about the lymphatic system as kind of an organ system, and now we're going to get into the immune system, which is housed within the lymphatic system. The immune system works on a fairly simple concept. It's you're either with us or you're against us. And if you're against us, then generally we need to kill you. So it's fairly black and white, and is all based on the concept of foreign versus self. This is a functional system, not an organ system. The organ system that houses this is the lymphatic system. There's two arms to the immune system that are going to work simultaneously with each other. The first is what's called innate or non-specific. Innate is referring to the fact that these are essentially present at birth and never really change their approach to whatever it is that they're targeting. Non-specific refers to the fact that they will treat everything the same. So I'll give you an example that we're going to talk about here in a second, which is a fever. So you are capable of having a fever at birth, and that fever doesn't get any better as you age. So the second or third or fourth time you see a given cold virus, the fever or the flu or whatever, the fever isn't going to get any better. Non-specific, let's think about something else we're going to talk about here, inflammation. So if I step on a rusty nail, I'm going to get inflammation in my foot. But if I hit my foot with a hammer, I'm also going to get inflammation. So it's very non-specific. It's going to treat both of those scenarios the same way. Classically, the immune system is described as a first, second, and third line of defense. I don't love that because it indicates that they are sequential when in fact they are simultaneous. So while we will refer to first, second, and third lines, please make a note that these are overlapping each other and all happening at the same time. So the first and second lines are going to be in this innate or non-specific arm. The third line of defense is going to be in what's called the adaptive or specific side of things. Adaptive means that it's going to get better. So it's going to learn and actually adapt its response to a given antigen. And therefore, it also has to be specific because it has to remember that it has seen this particular antigen before if it's going to get better at dealing with it. Within this third line of defense, we have then two arms there, which are called humoral immunity and cellular immunity. So here we can kind of divide this out. So within the innate defenses, the surface barriers are what are termed the first line of defense. The second line are internal but non-specific defenses like inflammation and fever. And then the third line consists of the adaptive defenses, which are going to consist of humoral immunity and cellular immunity. So we're going to start with the first, talking about skin. So your skin is a very effective barrier to pathogen entry into your body. It does a very, very good job. So you should be thinking about what type of tissue the epidermis is, and you should be thinking, well, it's protective. So hopefully you have spun those wheels and you've come up with stratified squamous epithelium, which is known for being protective. And in the skin version, it's keratinized so that the top layers are actually dead. So your skin, in addition to just being a good physical barrier, has an acid mantle. So we've got secretions that are slightly acidic. They're antibacterial. They're going to make it a not pleasant place for your non-normal microflora to hang out. We also have all that keratin in there that is going to be resistant to acids and bases, many bacterial enzymes, and different toxins. So the fact that those cells up at the surface are filled with keratin is going to help out quite a bit. Also in that first line are going to be the internal barriers, which are the intact mucous membranes. So again, what type of tissue do we find lining the oral cavity, the vagina, the anus, things like that? So if we think about what we're going to see here, in addition to just the effective barrier of stratified squamous epithelium, we're going to have things like mucus that's very sticky. We're going to have hairs. So a good example is your nasal hairs that are covered in that sticky mucus. They're going to help trap things that you're breathing. Cilia is going to be associated with certain types of epithelium, particularly in the respiratory system. It's going to help move that mucus out of the body. We've got some areas where we have very low pHs. So in the stomach, we've got a pH of about 1 to 2. Plus, we've got a lot of proteolytic enzymes. So anything you swallow is likely not going to make it through there. The vagina has an acid mantle that is going to help inhibit bacterial and fungal growth during the reproductive years of the female's life. Tears and saliva are going to help kind of cleanse, and there's kind of a flow rate. You're constantly secreting tears, you're constantly secreting saliva. We've got that sort of generic enzyme called lysosome, which is just sort of a generic antimicrobial enzyme. Urine is, again, a little bit acidic, and because, again, there's a flow rate, there's going to be this periodic cleansing that's going to occur. So even along the inside, that first line of defense is going to be a pretty effective barrier for us. Now, it's not perfect. So then we have to move into the second line of defense and see, okay, well, what do we have going on under the surface? And we're going to start with all the cells. So we've looked at these. We've seen these before. We know that these are our leukocytes. So we kind of introduced these in the blood chapter, and now we're going to talk about them just a little bit more. So we're going to start with the neutrophil. So neutrophils basically are kind of wandering around, and they are basically going to eat a lot of bacteria. So they are phagocytic, and they primarily eat bacteria, which we've talked about before. Their granules, though, are going to have a lot of chemicals in them that are going to help create what's called a killing zone around them. So not only can they eat something and kill it, they can also create an area surrounding themselves that's basically going to be filled with hydrogen peroxide and hypochlorite, which is basically what you would find in bleach. And so these surround the neutrophil and are going to create this layer of killing surrounding each cell. And so that's going to dramatically increase the amount of bacteria that a neutrophil could kill if it's not relying solely on being able to eat all of them. All right, the eosinophils. So we talked a little bit about these. We talked about the concept of the helminth infections, the idea that these are phagocytic, but they generally degranulate and begin to break down a parasitic worm externally and then bring the pieces in. In addition to that, they are also going to help out the basophils, which we're going to talk about next. So they have a role in activating and promoting the function of your basophils and then mast cells as well. Interestingly, they, in addition to promoting, they also are going to release an enzyme that inhibits the inflammatory response. It inhibits histamine and inhibits inflammation, which is going to help kind of pump the brakes once all of this gets going, because an overreaction of the immune system is just as detrimental as an underreaction. All right, the basophils, we talked about these guys. We talked about how you got to be really careful with them. We don't have very many of them because they have very, very potent messengers. So we talked about the histamine and heparin. So histamine is an inflammatory vasodilator, so it's going to help increase blood flow to an area. Heparin is an anticoagulant. It's going to make sure that no blood clots that would impede the flow. These are also going to release leukotrienes, which are going to help, sort of going backwards, they're going to help the action of neutrophils and eosinophils. So you're starting to get this concept of overlapping that I'm talking about, that nothing in the immune system is working on its own. Eosinophils help out the basophils, and then the basophils also help out the eosinophils. Now, remember when basophils migrate out of the tissues, they will be termed a mast cell, and they have a very similar idea going on. Now, the lymphocytes, basically we have B cells, T cells, and natural killer cells. These, we're going to be looking at all three of these populations of cells in a minute, so we won't get into that too far here. And then lastly, we've got our monocytes, so the biggest of the cells. These can migrate out to become macrophages. We know that these are definitely going to deal with viral infections, chronic bacterial infections. They are going to play a role in inflammation. And so we're going to see a huge role for particularly the macrophages as we go into this concept of the second line of defense, because we're going to find out that pretty much all of the other components of the immune system end up relying on phagocytosis. They can do something, they have their effect, but then what has to happen after that is that the phagocytes have to move in and get rid of whatever it is that they killed. So let's talk about that for a second. The primary phagocytes are neutrophils and macrophages. However, monocytes can do this in the blood, and then we also have eosinophils. But primarily when we talk about phagocytes, we're talking about neutrophils and macrophages. The macrophages in particular come in free and fixed varieties. Some macrophages simply wander and look for things, but others are fixed populations found in specific places. The dust cells in the alveoli that we're going to talk about, the Kupffer cells or hepatic macrophages that are going to be found in the liver, the microglia that we talked about in the central nervous system. That's an example of a fixed population of macrophage. Now, phagocytes do need to be told that it is okay to eat something. Otherwise, they may wander around and eat some things that we don't want them to do that with. So, we want them to be able to recognize what's appropriate and what is not appropriate. And the process that we use to do this is called opsonization. We're going to tag anything we want eaten with something that is going to draw the macrophage in. So you want to think about the process of opsonization. So think about something that you really like to eat, okay? I really like tater tots, so this is always what I use as an example. I really like tater tots, but, plain tater tots, I mean, they're okay, but they're not great. But if you start topping the tater tots with, like, bacon and cheese, I'm going to be all over that. So the bacon and the cheese are drawing me into these tater tots and making it absolutely impossible for me to just walk by them. Same concept of opsonization. We are going to put things on the surface of viruses and bacteria and cells that need killed that are going to draw in your phagocytes and make it almost impossible that they would miss it, that they would just walk by it. Now, of course, we're not using actual bacon and cheese here. We're going to use things like antibodies and complement proteins. And what they do is they just function as a big, neon light that says, hey, you're going to want to come eat this. This is really good. It's very tasty. You're going to want to come in here and you're going to want to eat it. So that's how we're able to have these populations of cells that go around and eat things, but also have them be regulated. So we're going to tell them what we want them to eat via opsonization. Now, once they do get to eating something and they are able to adhere to whatever it is that we're dealing with here, looks like a rod-shaped bacteria, what happens is they're going to use endocytosis. So we talked about this idea that not every cell can do this, but these can. And they're going to form this vesicle when the membrane pinches off up here. That's going to fuse with a lysosome that is then chock filled with what are called acid hydrolases, a whole population of enzymes that break down pretty much every type of molecule you could think of. And so, you're going to chop this up and then you're going to use exocytosis to spit out the remnants. So you have to tell them to do this. And a lot of times it's something small, they can handle it, and they just spit out the rest. Sometimes they engulf something so big that the cell actually has to die along with whatever it engulfed. And that's basically what we see when we see pus. So pus is basically dead macrophages and neutrophils that have brought so much in that they have died along with their cargo. Okay, we're going to talk about two sets of antimicrobial proteins that fall into this second line of defense. Again, innate and nonspecific. So we're going to start with what are called interferons. Interferons are a defense against viruses. So quick little view on viruses. A virus sitting on the table can't do anything. So viruses are not classified as either living or non-living. They straddle the line there. And they are really utterly useless until they gain access to a host cell. Then they hijack the host cell and turn it into a virus-making factory. So all of the host cell machinery is going to get sort of shut down on all of its normal processes, and we're going to just start making new viruses. So that's kind of how a virus works. So when a virus enters and we have the presence of viral DNA or RNA, it is going to trigger the gene for interferon to switch on. The very last thing this cell is going to do under its own processes is it's going to go through transcription translation of this gene, and it's going to release interferons from this cell. Now, this cell is done for. We cannot help it. It is over. It is going to die. This virus is going to kill it. The goal of interferon is to save the neighboring cells. So cells have interferon receptors, and when a neighboring cell releases interferon and it binds, it's going to trigger this second cell to make antiviral proteins. And that is going to help this second cell from being infected by the virus. So interferon cannot save the original cell, but it can save the neighborhood. Okay, it's a signal that's being sent out in a paracrine fashion to tell the neighboring cells, hey, I'm infected with a virus. I'm about to try to blow up the neighborhood. I'm super sorry about this. There's nothing I can do about it, but you can try to save yourselves. That's the goal of interferon. Now, complement proteins are a little bit more complicated. they function in a cascade effect. So similar to blood clotting, we talked about this idea of a cascade in that we would have all of the components in the blood at all times in an inactive form so that we could activate this very quickly. Okay, so there's three ways to activate complement. Classical is antibody driven. So you've got to have an antibody antigen complex that drives the process to begin. Alternative is basically almost like kind of like a spontaneous reaction that usually occurs as opposed to sort of positive action, it occurs through negative action. It occurs if there is not something to block it on the pathogen. So that's why we kind of call it sort of a spontaneous, but it is antibody independent. It does not require antibodies. And then the lectin pathway is going to be activated when lectin binds certain sugars that are really only found on pathogens. They're not a combination of sugars that we would find on our cells, therefore it's going to be registered as foreign. The lectin pathway is also antibody independent. Now, doesn't matter how you start it, you're going to end up with complement protein 3 dissociating into C3a and C3b. Okay. Now, you don't need to worry about all of the steps here. Okay, let's just go through the different things that can happen once we get this going. First of all, we're going to help out inflammation. Okay, so we're going to have multiple second line of defense mechanisms that are all going to interact with each other. Inflammation works better when complement has been activated. You've got immune complexes, okay, so the clearance of these, particularly when you're dealing with red blood cells, so those ABO transfusion reactions, so they would get cleared via the liver and the spleen. Here we are using complement as opsonization, so bacon and cheese. So when we tag things with the C3b protein, it is going to draw in your macrophages, which is going to help out phagocytosis. So again, phagocytosis works better when complement has been activated. And then, this one's kind of cool, it's going to cause the cell to blow up because it is going to form what's called a membrane attack complex. So here you get all of these different complement proteins that actually insert themselves into the membrane of the target cell, and they form a pore. And this is going to allow the contents of the pathogenic cell to begin to leak out, and that cell is going to die because it no longer has a functioning membrane. So these are called MACs, or membrane attack complexes. So 3 ways to start and 4 things that can happen. Okay, so definitely a little bit more complex than our interferons, but you're going to have a wider range of things that could be utilizing complement as opposed to just a virus. All right, natural killer cells technically come from the lymphoid line of cells, but they don't function in the third line of defense like the B cells and the T cells. They function in the second line of defense. These cells literally just wander the body and they look for anything that looks a little weird, a little off, a little funny. And by looks, remember what we're doing is we're looking at this concept of what is on the surface of the cell, what markers, what proteins, what sugars, what is on the surface of the cell. And when they find something, they immediately kill it. And so here you can see this is your little natural killer cell with its perforin gun, and here, you know, he's shooting him, okay? Now, if you want to see what it really looks like, it's down here, but I think these are way cuter. So what actually happens is, again, the natural killer cell is going to release perforins that in exactly the same way, just a different protein, are going to form a pore in the target cell, just like the membrane attack complex we just looked at. Once that pore is formed, it is going to throw in some granzymes that are kind of like grenades into the cell that is going to cause this cell to undergo apoptosis. And then here comes our macrophage in here to eat it up and take care of it. So that's two different things that we've seen in the immune system that function simply by poking holes in other cells. So when you think about the importance of a plasma membrane, think about that. And we're not even done. We will see one other case where the wildly complex human immune system functions by poking holes in things. All right, a fever is going to fall into our nonspecific defenses, which again, doesn't matter if you have a bacterial infection or a viral infection, your fever is going to function the same. Fevers start with chemicals called pyrogens, and pyrogens are going to target the hypothalamus because that is where we set our body temperature. So here we're looking at the green line is the set temperature. So this is the temperature your hypothalamus has set, while the red line is your actual body temperature. So normally the hypothalamus is hanging out around 37 and we are oscillating around 37. When pyrogens show up, the hypothalamus is going to turn the thermostat up. Okay, so instead of being set at 37 degrees Celsius, it's up at 39. And that is going to trigger what's called the onset phase of a fever. Remember that your sense of temperature is based on how far away you are from the set point. Whether you feel cold or warm is based on what direction are you going away from the set point. So during the onset of a fever, you're going to feel cold and clammy because your body's telling you you should be at 39 and you're down here at 37. So you're going to do things that are going to allow you to gain temperature. You might be covered in blankets and you might turn the heat up. You might even get to the point where you start to shiver in order to generate heat. Once you get up to your new set point, you begin to oscillate around that, and this is called the stadium phase. So here, you're not going to feel nearly as miserable as you did here. Okay, this is by far the most miserable portion of a fever. Once you get up here and you begin to oscillate around this new set point, you will not feel nearly as cold. You may still feel sick because you're working on clearing whatever is causing this fever, but you're not going to have that cold and clammy feeling. Now, when the infection is gone, the hypothalamus is going to turn the heat down. It's going to bring our set point back down to 37, which is going to trigger you to go into the last phase of a fever, which is defervescence. Here you're going to feel very hot. This is the point where we would say that the fever breaks and you get sweaty and hot and, all of those blankets and things that you've piled on, they're off now, and you are trying to release heat to get back down to your set point. Then you begin to oscillate around it again, and you're good to go. Now, a fever in and of itself is not going to clear an infection. A fever is just a helper. It can help inflammation. So inflammation works better if you have a fever. It helps phagocytosis. But fevers are simply a helper. And that also means that, if you leave your fever alone and give it a chance to do what it's supposed to do, that you will, in all likelihood, clear an infection sooner than if you take Motrin or Tylenol or something like that to reduce your fever. Fevers are not a problem outside of three populations. If you're very young or very old, babies or the elderly, um, the hypothalamus can change your temperature, but it doesn't always do it fast, and it takes longer to go up and down. And so, you can keep rising and rising and rising before you realize you're past your set point. So, babies and elderly don't thermoregulate as quickly or as efficiently as, you know, a kid or an adult, somewhere in the middle portion of your life. So, a fever in a baby, a fever in somebody who's elderly could be an issue. The third population we would be concerned with is anyone who is immunocompromised. So, if fevers are simply a helper, okay, then someone who's immunocompromised is not going to have the rest of the immune system to kick in and clear whatever this is, which means the fever can become unregulated. So babies, elderly, immunocompromised, fevers are reason for concern. Outside of that, fevers are not really a big problem, and we don't need to fight them nearly as much as we do. Yes, they kind of make you miserable, particularly in the onset phase, but they are helping your immune system work. All right, and then that's going to lead us into inflammation, which inflammation is the biggie here. If there is one thing that's going to follow you through your medical career, it's going to be inflammation. That is because what is a normal response, the inflammatory response, can be triggered abnormally. And in fact, abnormal, unregulated inflammation is at the heart of most human disease, which is why, of all things that are going to follow you through your entire medical career, the concept of inflammation is going to be a big one. So we're not going to look at the pathological side. We're going to look at the normal inflammatory response as it functions as an immune piece. So the goal is to prevent the spread of anything that's damaging tissue, help get rid of the sort of debris and pathogens that come from tissue damage, alert the third line, the adaptive immune system, that they need to sort of move in here and start looking at this, and set the stage to repair the tissue. So none of that looks bad. These are not bad things. And so I really want you to distinguish what would be the normal inflammatory response from pathological inflammation. There are 4 cardinal signs of inflammation with a possible 5th. We've got redness, heat, swelling, and pain that are the 4 cardinal signs. The 5th one is loss of function. So that's right here. And loss of function is going to depend on both where the inflammation is and the degree of inflammation that we are dealing with. So if you hit yourself with a hammer on your elbow, you're probably going to lose function of the elbow for a period of time. But if you hit yourself with a hammer, right in the middle of your forearm, and it's not going to affect the wrist or the elbow, you're probably not going to lose any function. So first of all, location can matter. Second is going to be the level or the degree of inflammation. You can have a significant amount of inflammation in your liver before you actually begin to show signs of that, or there is any actual decrease in function. So both of those are going to matter when it comes into this concept of loss of function. So all inflammatory responses are going to start with some type of chemical alarm, which is generically called a chemokine. Okay, so there are immune cells and tissues that can release these chemokines that are then going to draw in the cells that are going to be needed. Now, this one's just funny. this is what it actually looks like. Now, you do not need to worry about this. I am putting it here just so that you can see all of the different chemokines involved and all of the different populations of cells that are involved here. So when we talk about the inflammatory response and then in your future classes get into the pathology of it, you come back to this and you can see, wow, this is a big concept, and there's a lot of moving parts to it. So we kind of talk about step 1A and 1B. Okay, they're happening simultaneously, so we can't really divide them into step 1 and 2, and they are equally important. Once the alarm has been set, the immediate response is going to be vasodilation and increased vascular permeability. Vasodilation is going to lead to hyperemia, which means more blood is going to come to the area. That is going to be responsible for the redness and heat that we see in our cardinal signs. And the increased vascular permeability is going to allow exudate to seep into your tissue. So the plasma component of blood moves out into the tissues. It's going to contain clotting factors that are going to help wall off this area of possible infection. Proteins, including the antibodies that we're going to get into a little bit later. And your immune cells are going to sort of rearrange their cytoskeleton and move through the vessel wall where it's been pulled open a little bit. The repair of this tissue is going to rely on all of this moving out into the tissue and kind of creating a scaffolding for the vessels and fibroblasts and things like that to come in and help fix this. We're also going to see a huge influx of the phagocytes. It's going to start with neutrophils. They are the first responder. They are there within like an hour. And then at somewhere between 10 to 12 hours, you're going to see them replaced with the macrophages. So here we can kind of get an idea of how this works. We've got the splinter here that is covered in bacteria, bad splinter, and all of these tissues are in fact damaged. So we've got this SOS that goes out from the damaged tissue. And so that is going to trigger our inflammatory response. We're going to get vasodilation with increased vascular permeability. You can see that we're going to pull these cells away from each other, the endothelial cells. That's going to allow these cells to move out through this process called diapedesis into the tissues, and then they are going to follow what's called a chemotactic gradient. So chemotaxis, this concept right here, is the concept where a cell moves up a chemical gradient. In other words, right here at the area of injury, there is a very high level of these chemicals that are drawing these cells in. Way over here, there aren't as many, which means that a cell would move in this direction as it goes up the chemotactic gradient. And then again, what are we going to do when we get there? We're going to rely on good old phagocytosis to start cleaning everything up.